Hello and welcome to the AutomAl 6000 Quickstart tutorial. My name is Håkon and I'm the creator of AutomAl. Now, AutomAl 6000 is a program for analyzing high-angle annular dark field scanning transmission electron microscopy images of precipitates in aluminum, magnesium, silicon, copper alloys. And the goal of this video today is to get you started with the software as fast as possible so that you get to try it out and see if this is something you can use. I will try to go through the things fairly without technical details, and we might make more topic videos on a later stage. But for now, I will try to get to the essence of how to use the software. To get AutomAl, you can go to automal.org. I will provide a link in the description. And here you will find some general information and guides and some links, but for now we're interested in the download section. And there is basically two options. You can either clone the source code from GitHub and run it locally. And as far as I know, this should be cross-platform, but I haven't tested it on anything else than Windows, so I can't really say for sure. And we have also tried to make an executable version, which you can download here, and this will only work for Windows. So you just so this will download a zip folder. So you just click the one with the highest version number, and you just download it. So once that has downloaded, you can just go ahead and extract the folder. It's a zip folder, and inside it you will find a file called automal6000.exe, and this can be used to run the program. And you can, of course, this folder, you can place it wherever you want, and you can make a shortcut to this executable. And to test it, we just click it, and yes, you might get something like that. You have to click more info and run anyway. It is not a virus, I promise. And you notice it might take some time, but it's important that you don't click several times while waiting, because it's unpacking some files in the background, so it might take some time. So here is the interface, and so now we know it's working, and yeah. So first, just a quick tour of the interface. On the left is the control window, which has several group boxes with buttons and readouts and options. It might seem like a lot here, but not all of it is important. We'll only be looking at the things you'll actually need to get started today. And these can be collapsed or expanded by double clicking on the group title. Then in the middle is the view box. It has several tabs which will display different aspects of the information. We will see that later. And on the right there is a terminal output. Sometimes this output will seem sensible, but most of the time I think it might not seem sensible to most, but it's there for mostly debugging purposes. And then on the bottom there is a system bar which indicates if the software is working or ready, or yeah. So to begin using AutomAl, we first have to import some data, and depending on which column detection method you would like to use, there are two options as far as I know. There's some slight differences. So the first option is to use AutomAl 6000 column detection, and I will show this afterwards. The other possibility is another software called AutomAl by Magnus Nore at NTNU, which some of you may be familiar with. I will provide a link in the description as well. And what you can do there is that you do the column detection in AutomAl, and then you save the AutomAl file as a HDF5 file format, I believe it is, and then you can import it here to get both the images and the column positions here. But for the other option, if you want to use AutomAl's column detection, which is a bit less sophisticated than the atom map, it's a basic center of mass method. What is important that you do first is that the image has been noise filtered, which means that, let's say you get a DM3 image from the microscope, you first have to open it in some, let's say, digital micrography suite or something, and then you have to do the FFT and then an appropriate low pass filter, and then inverse FFT again, and then save that result as a DM3 file. And once you have done that, you can go ahead and import it here, which I will do. And actually, with AutomAl, there is a sample included, so I will use this today. And just open, and it will take a little bit of time, and it will say here that it is upsampling the image. Yeah, this is a pop-up box with species dictionary. You can just cancel this for now. I will explain that later. Yeah, so as I was saying, it will say that it has upsampled the image if it finds that the scale is such that it needs to do that. And that's for the reason for that has to do with the column detection method. But just it will be automatic, but just keep it in mind that the images you are exporting later might be twice the size of your original image in terms of width and height. Yeah, so just keep that in mind. And it will also say here if this upsampling has been done. So if it's false here, it should be the same. Yeah. So now we have opened our image. We are now looking at the raw image tab, and what you can do is you can use shift and mouse wheel to zoom in and out, and you mouse wheel for up and down, and alt mouse wheel to go left and right. Yeah, and one final thing for importing is that this is not professional software. It's kind of I was learning how to make software of this scale at the same time as I was doing my master's thesis, so there might be problems here and there, which means that it is important that you save often. And to do that, you just click save here, and I will call it sample demo, and AutomAl files do not have any extension, so just sample demo and no dot anything, and save. So now that we have successfully imported our image, we want to go ahead and do column detection. And the method is unfortunately not fully automatic. It will require you to guess a specific value, and this is called the detection threshold value. This is a number between 0 and 1 and will indicate kind of the darkest column which it will pick up. And this will be different between images. This image has copper, which have very bright contrast. This will shift the contrast of the other columns down relatively. And let's say you had a pure beta double prime with no copper, then all the columns would be brighter. So for this one with copper, or like a bright column, we want to somewhere between 0.2 and 0.25, and for non-copper images maybe you can guess something like 0.3. And I will guess a little high here now on purpose to show you kind of how you would go about to narrow in on a sensible value. So once you have made a guess, it's not important really that the guess is correct, because the method will continue from where it left off if you select a new threshold value and then restart it. So you will be able to kind of narrow it in. So I will press start, and I will just select the threshold. That's the search type. and then press OK. And it will say here that it's starting column detection, and down on the system bar it will say that it is working. And this might take somewhere, well, for this particular image I expect it to take maybe like 3 minutes perhaps. Yeah, so I will come back when that is done. So now it says here on the right that the column detection is complete, but you will see nothing because we are in the raw image tab. But if you go to atomic positions, you'll now see the locations of the columns it has detected. And just a quick tip, if you want to hop between tabs and you want to kind of, let's say you are here in this image and this one is like there, and you want it to kind of align, then you can just click this lock views and it will lock to the viewport of the current tab. So if I wanted this to be what I want to look at right now, I just press lock view, and if I go to here now, it should be approximately the same position. And you can go back and forth with the tabs with Z and X, which I find useful often. And you can also not have them locked. So when it's locked, then if I change the position here, then it should be changed here as well. But you can also individually align. So let's say I'm interested in this area and I want all other tabs to align to this area. I can press E, I believe it is, and then yeah, all tabs should be aligned to that area. So that's kind of useful, I think. So yeah, now we want to look at the column positions, and we see here that indeed it found some columns, but there is a lot of here that it did not pick up on, and that is because if there are columns that are left undetected, then we have guessed too high. The other thing that can happen is that it will find columns in between other columns, like it will find columns that are not there. That means you have guessed too low. So what I want to do now is that seeing that this was too high, I want to set the threshold value again, something lower, let's say 2.5. No spoilers. Okay, and then I press start and just keep this threshold the same as before, and it will not redo the column. It will start from where it left off, as I mentioned, which means that now it will not take so much time. So as you get closer and closer, the method is faster and faster, and it will take, like, in the end it will just be seconds. So I'm hoping to get it up now fairly quickly. And you know, this is a bit finicky. I think it's a bit annoying, which is why you might find it useful to use, for instance, AutomAl. So there we go. It says column detection complete, and this result has to be controlled. These edge cases here are not important, but it looks quite okay now. I cannot see any errors. I know I've seen this image a lot of times, so I know there's a particularly weak silicon here, but I will show you how you can, let's say it's one missing or there's two, or like it's not many, then you can manually set them. Which is what I will do now. So if I go down here to selected column, there is an option here which is, or a button that says new. So I will click that one, and that will insert a column in the middle of the image and enable something called move mode. So when move mode is enabled, I can actually move around on this column, and this is a little bit buggy, but because if I do like this and then I want to move it again, it kind of hops back, and I was never able to fix that bug. But what you do is you take it to approximately where you want it, So let's say here, and then you can use WASD to kind of move it at pixel increments. So I'll just use that to kind of position it like that. Yeah, and then to save the location and exit move mode, you can either click accept here, or you can just press enter. I believe it is it? I don't remember the keys right now, but all the keys, overview all the keys can be found on the web page for later. So that is our columns. Seems good. And yeah, and you might have cases where it's like a bit ambiguous, and like that's kind of up to you what you want to do. I think this is the most correct here. So, and yeah, so now as always, very important to save. And what I would like to do, is to go ahead and just take the name of the imported file, and then I just add the stage we're at. So I'll call this columns. And the reason I do this is because there is no undo function in AutomAl. So if something goes wrong, and then you cannot go back, kind of. So save often and save at multiple stages is my suggestion. Yes. So now that we have successfully done column detection, we get to kind of the real meat and bread of AutomAl, which is the column characterization, which it is called. And this is an algorithm that looks at these column positions, and it tries to figure out both the species, the atomic species of each column, and also its set location, which like its height, like its position along our viewing direction here. And the way it does this is fairly involved. It's not something I can explain very quickly, but if you are interested in technical details, we are working on a paper that hopefully should be out very soon. And I will link that in the description as well and on the webpage once it's available. But today I will just talk about the most basic things you need to know to be able to run this algorithm. And I think the most important thing is what I have termed the species dictionary. So if you go to project, there will be a button called species dict for dictionary. And this is the same pop-up that came once you imported the image. And what this means is that because of course we want to categorize each atomic column into each atomic species which we have present in the image. And this is all nice and fine, but it turns out that for this to work well, then there are actually subcategories. Like, let's look at silicon here. So this silicon here says silicon 1. This is connected to this one, silicon, of course. I think it should say, maps to atomic species silicon. And it has a description also. It says that this is beta double prime silicon. And if you look at the second one, it says that it is Q prime silicon. And what is going on here is that this silicon 2, which is nearest neighbor to the copper in Q prime, actually has a different symmetry tendency than silicon that appears in interfaces or in beta double prime. So for the method to work, it uses like a part of the method uses a statistical model. So this subdivision helps in that. And so which categories you want present depends on what kind of what is in the image. If you are looking at a Q prime or beta double prime or disordered, or what do you expect to find. So if I look at this silicon 3, it says here interstitial beta prime silicon. And actually the method doesn't work on beta prime because of the interstitial silicon position. But there is no beta prime here, only beta double prime, so we can just go ahead and remove it like this. You can also add your new ones. You can add more species and connect them, and you can play around, but for the most part the thing I end up here will be the thing you need in the 6000 alloys. So here you have copper 1 is the 3-fold symmetry copper, and then the copper 2 is a 4-fold symmetry copper. Now the 4-fold symmetry copper doesn't appear often, and it's definitely not anyone here, so I will remove that one. Aluminium 1 is the matrix aluminium, and element 2 is precipitate aluminium. We won't want those. The 2 magnesium ones are, it's something to do with the angles, but keep both of these if there are magnesium present. And then you have 2, so UN stands for unknown, so it's kind of like a unknown label. We only need the 1 unknown species, and this one is for vacant and void column. I don't think there are anyone here, so I'll remove that one. And this list here is what you will typically be using, but this kind of shows you kind of the possibilities here, and yeah. So I will click apply, and if you ever make a mistake, you can always restore the defaults, and yeah, so you get the point. And so that's the first thing you would kind of have to figure out. The second thing the algorithm needs is that it needs like a starting point, which is known to be in the matrix. And this means we have to first manually set one single column. I will choose this one, and it's important that it is in the matrix and not in the precipitate. And I want to set the species of this column to aluminium. Now to go ahead and do that, there are several ways. We can go down to selected column, and we want to set the atomic species to aluminium like that. Now the next thing we may want to look at is the overlay composition tab. So if I go to then, so this teal color or whatever it is, it's indicates selected columns. It's a bit weird, but anyway. So we see now that this one has turned green, and if I zoom out to see the legend, we see that this is the aluminium. And I will discuss this overlay a bit more later. There are some things you can do, but for now what we need is a single aluminium column like that. And the way we did that was that we selected the column, and we went to select column control area, and we set the atomic species manually. But you can also set it with buttons. So 1 is silicon, I believe, 2 is copper, 3 is aluminium, 4 is magnesium, and that will be very useful later when you want to move a bit quickly. So we can do like this, but actually I don't want them. I want them to be unknown for now. And set like that. So that is kind of the preparation stages. So first is to set the correct species dictionary, and then select a column in the matrix, set it to aluminium, and then keep this column selected. So this has to be the selected one. And then you can go to column characterization, and you hit start. and this will give you a pop-up box with a Dropbox menu, which has like a tons of options here. But what you want is the top one, the default one, the 0 full column characterization algorithm. And then just OK to run it, which I will do now. And yeah, it will be a lot of this and that in the output, and it will say here that it's analyzing, and this might take some time. This image will be fairly quick. I expect maybe 4 or 5 minutes tops. But for larger images, this grows quickly. So it's and by large, I mean large in terms of the number of columns in the image. So for a huge, let's say you have a huge dispersoid something, then it might take like half an hour in the worst case. But for these types of images, typically around 5 minutes. So here we have the result. It says here full column characterization complete. It took for this image it took 90 seconds. Yeah, here you can see the result in the overlay composition, and here you can see the atomic species indicated by color, and their relative set height indicated by hollow or filled symbology. So we see we have in the matrix, it looks like they got all the heights correct, and I believe it is fairly correct here too. And yeah, so this is a good result, but there will always be it will never do a perfect job, so you have to help it from this stage. And so let's say I wanted to check that this is a good result, it is correct. I would have to, okay, is this correct? And I would have, okay, is this correct? Blah, blah, blah. And, it would take forever. So to remedy this, like there's something called atomic graphs, which we have been using to run this important part of the algorithm. It kind of maps the relationship between the columns, which is important information. And this is mostly for the algorithm, but it turns out to be a very useful tool to review the results as well, because just looking at this, even though it looks kind of okay, it's for bigger precipitates, you can't sit and check every single one. So what but before I show you the atomic graphs, I wanted to point out a fun fact here, and that is that if you see here, we have 2 columns that are still marked as unknown, and that is because we were fiddling around with them, and I was manually setting their species to unknown. And when you manually set a column, the algorithm is set up to not change the species of that column. So yeah, that's just something to keep in mind. So I will set this to aluminium now by pressing 3 while they are selected. Alright, so atomic graphs in a second. So before I show you the atomic graphs, remember, as always, we have to save. And this time I will call it result. Yeah, so that we always can go back here without having to rerun the algorithm. And yeah, so if we now go to atomic graph, then I will zoom out a little bit. This is where your knowledge of 6000 alloys is gonna come into play. And this is a visual representation of the structure, and it's in my experience when showing this to colleagues, then it's a bit steep learning curve, and it will take some getting used to. But once you do get used to it, then this will be a very quick process, because you'll be able to see things like instantly, and just correct it, and then you have your result. But I'm going to try to take it very slowly here, and I'll try to explain what all this means. So this atomic graph is actually a digraph, which means that the edges between the vertices kind of have direction. And you see this, for instance, here there's an arrowhead pointing this way. This means that this edge is pointing this way. All the other edges, or most other edges, are black or have arrowheads both ways. When they are just a thin black line, it means it has two edges, one going this way and one going this way. So it's a symmetric arc. And symmetric arcs is what we want. But we see here that there are some problems. There are some red and blue, and this we don't want. Because what this means is that this atomic column thinks that this is one of its opposite plane nearest neighbors. And by that I mean that you see here you have the different colors, white and black. That is the two different set heights. And by opposite plane nearest neighbor, I mean that these two are nearest neighbors, but they are on opposite planes. This one and this one is also nearest neighbors. Actually, let me zoom in a little bit. This one and this one are also nearest neighbors, but they are nearest neighbors on the same plane. So what we are most interested in is the opposite plane nearest neighbors, because they and so what you can see here is that this thinks that this is one of its opposite plane nearest neighbor, which is not correct, as we can see. In fact, if we look at it, then we can see that actually this one is one of its opposite plane nearest neighbors. And I will show you in a bit how you can actually manually edit this information. But first I wanted to talk a little bit about like the structural principles that are in these types of precipitates and systems. And these principles are the reason why this algorithm works, which means that if you don't have these principles, then this structure couldn't have I wouldn't have known about the method for how to determine the structure without any structural principles. And the principles are in a very like easy, simplified way. They are that the number of opposite plane nearest neighbor to each column is specific to atomic species, or more or less. And in the aluminium, magnesium, silicon, copper system, then silicon and copper takes on 3 opposite plane nearest neighbors. So you see here, this is either a silicon or a copper. I don't know which one it is. Looking at this graph, I just see that it has 3, and then I know it should be silicon and copper. But to distinguish between silicon and copper, we of course rely on the on the contrast brightness. So if I now align the views on this one and I press set to go to the left, excuse me, to look at the raw image, we see that this is in fact a silicon. So if we now go back to the overlay, we see that indeed this is labeled as a silicon, which is a correct label. And so that's for any position that has 3 opposite plane nearest neighbors. And we can see from this atomic graph that the software knows that this has 3 opposite plane nearest neighbor because it has 3 edges going out and into the column. So just looking at this graph, we can see here we have a silicon or a copper. Now for aluminium, it takes on 4 opposite plane nearest neighbors. And again, we can see here that that the program has correctly selected this as aluminium because it has 4 symmetric arcs. And then finally, magnesium takes on 5 opposite plane nearest neighbors. We see one here. There's a 1, 2, 3, 4, 5. And again, we can see that this is correct because it has 5 symmetric arcs associated with it. So this means that, okay, looking at this, I don't have to actually go and check the label to see if this is in fact magnesium. I know that because there is only magnesium who has 5 edges. And so this is the kind of the principles that you have to rely on. And if we now move on to kind of the problem area here, we want to see what is wrong. First of all, this is pointing to a same plane neighbor, which is wrong. And but we can see how many extending arcs are there. There is one here because of the symmetric arc. You remember it goes both ways. So we had one arc extending out here. There is one arc extending out there, and it has one there and one there. So this has 4 arcs extending out from it. This means that the software believes that this is aluminium, which I think is correct. But the structural relationship information is wrong. So in this case, we wouldn't, and of course, this I realize this might be a bit difficult to see like off the bat, but when you think, if you get to think a little bit about it, you will understand. And in this case, we won't have to change the species, but we want to change the structural information to be correct. And to do that, there is on the left here, there on the panel, there is under the atomic graph group, there is an option called enable permute mode. And you can either click on that or press spacebar when you are in the atomic graph tab. And once you have done that, the program expects you to select 3 columns in a specific order. And this order is, let's say we want to change this arc here to not point to this one, but we want it to point to this one. And the way we would do that is we select first the pivot point, which is this one. Then we select the old neighbor, which is this one. And then we select the new one we rather would like it to point to, which is this one. So if I do this now, enable permute mode, and then I click on the pivot on the old head and the new head, this will now point to this one. And then very important to remember to disable permute mode once you're done. So now this is correct, but here is our next problem. This one is pointing to this one, but this is not pointing back, because if it were pointing back, then this arc would be symmetric and the line would be black. And the problem is that this one has 4 extending arcs, but it should have 5. So this is mislabeled as aluminium when in fact it is magnesium. This means we have to manually change the species, and as you know, you can do this from this menu here, but that is slow, so I would use the numerical button 4, and 4 was magnesium. And again, an overview of all the buttons can be found on the web page. So if I press 4, this line now suddenly becomes black, because now that the software thinks that this is magnesium, this one gets an extra arc to extend. and this arc becomes symmetric. And now you start to see like how this can start to go pretty fast. Like we see this one is pointing here, which is the same plane, which is wrong. It should be pointing to this one. And so if I press spacebar, pivot point, alt point, new point. Here we have the same. This is pointing there, should be pointing there. So I press the pivot, alt, new, and then I disable permit mode. So that's kind of how you would go about, because the big the benefit of this atomic graph is that all the problem areas, this is an area where the algorithm had problems figuring out the structure, but it is easy to identify these areas and fix them manually once you are familiar with these atomic graphs and kind of the interface. It gets some getting used to, but it should be fine. And by the way, blue lines means that both of them agree that they are nearest neighbors, but they are of course on the same plane, so that should not occur. And the kind of the thesis of this method or algorithm is that in areas where all the arcs are symmetric, like the area we are looking at now, you should not need to check it. Like this should be all correct. I have seen very rarely any counter examples to this where all the arcs are symmetric, but the structural information or the species information is actually wrong. And this can happen in some images where there are like big smears or very ambiguous positions or like very disordered or something like that. But in like highly ordered precipitates like this one, if you use and it looks kind of sensible like this, then it is usually correct, with the one exception that we can't see the difference between copper and silicon. Now, so the final step in the review of the result would be to check that all the copper are labeled as copper. And of course you would clear out this area first, but I'm just gonna go ahead now and go here, show you how I would do that. So if I press E to align the views, I see here that okay, this is copper, 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 and all the rest should be silicon. So I see here I have a copper here, but this should in fact be silicon. Or yeah, you can see why it thinks this is copper, but as this structure looks like, this should most likely be silicon. So I'll press 1 to change that to a silicon. And you have 3 here, that looks correct, and 2 here, that looks correct. So now all our silicon and copper should be fine. And so this is all fine and dandy, but eventually you will come across something like this. And this is in fact an exception from the principles we were talking about earlier, because you see this one here is correctly assuming this to be its opposite plane nearest neighbor, but this is not reciprocated. So, and why? Because this magnesium allegedly has 1, 2, 3, 4, 5, 6 opposite plane nearest neighbors, but I said that magnesium should have 5. But these kinds of 6 opposite plane nearest neighbor magnesium do sometimes occur in 6000 allos, which is one of the interesting results that we noted in this work, because looking at these graphs for many images, we see that this actually happens sometimes. Yeah, and yeah, one final piece of the puzzle is up here. We see that this one should point there, this one should point there, this one should point, not sure actually. So this is again, this is like a 6 opposite plane nearest neighbor magnesium kind of, but if you go back to the actual image, we see that this was the area which is a bit like, I don't know really what is going on here, and this is the situation kind of where your expertise have to like fill in the blanks, so to speak. But for now, I think that my guess would be that this is actually silicon, and this one is aluminium, and this is also silicon. And now you saw I changed the species to silicon, which means that it lost an arc, but it lost the wrong one. It should have lost this one. So this, we don't know the order in which it has arranged its neighbors, so we would have to correct that by same as usual, like that. And this arc, in my opinion, should just stay as it is to indicate that this is a deviation from the principles. And so that is kind of the how you would go about reviewing the atomic graph. And sometimes it can be harder than this or more ambiguous than this, but mostly it's should be fairly easy. So one final thing I would like to mention about these atomic graphs in this video is that we talked about the principle basically being assigning atomic species to a number of opposite plane nearest neighbors. And a consequence of these principles, combined with the fact that there are only 2 possible heights, and this is actually a bipartite graph, is that every mesh, and by mesh I mean an area enclosed by arcs, every mesh should have 4 vertices, and which means that every mesh should be 4-sided. And this is actually very useful when you are looking at problem areas. So something typical that you might come across that might seem confusing when you are not so familiar. I'm gonna create this situation artificially. Something like this can happen. It doesn't happen often, but it can happen. And when you start looking at this, it's like it can be especially if it's more complicated than this, it can be a bit hard to kind of know where to start untangling the mess. But it is very useful to keep in mind that triangles should be avoided, and we start at like one edge and we see here is 4, so that is all good. This should really be like a 4-sided one. So I'm just gonna go ahead and put this back, and you see now I have 4 sides, which is good. So I know that this edge should be the correct one, and I have an available connection here because this is again a 3-sided mesh, so I can go ahead and move that there. And to remove this completely and make a 4-sided mesh, I can go this there and this there. And so that's kind of important tool. And of course you may want to check that actually every single mesh is 4-sided if it is like a coherent structure you're looking at. And AutomAl also has a tool for that, and it's something called show mesh vertex order here in the atomic graph tab. And I can click it and really nothing happens because it first has to build like the mesh list kind of thing internally, because it's kind of slow, so it doesn't do it automatically. But if you need to and want to, you can press this button called refresh mesh, and there will be some output and it will work, and it will take a little bit of time, maybe 30 seconds or a minute, I don't fully recall. But once that is complete, you can enable this box called show mesh vertex order, and it will place a number in each mesh with its number of vertices, and the ones that are not 4 will stand out in red font. So this can sometimes be also another useful tool if there are problems in the kind of in the graph. So now it says here that the meshes are mapped, which means that now I can press the show mesh vertex order, and you will see that every mesh gets labeled with a number. And like now there are no red numbers anywhere, so this should all be fine. But keep in mind that this doesn't update continually. It has to be refreshed with this button every time a change is made. But it's just like a last thing to check before you go ahead, which we will do now. This overlay here now should be the correct overlay for this precipitate, in at least according to my knowledge. And as you can see, to summarize, it's our knowledge about these principles, structural principles, will have to be used in conjunction with the algorithm, because, as I said, there will always be some problems that will need some attention. But at least atomic graph tool like we have here is very helpful to go through that quickly. And of course, now we have spent a lot of time talking about this, but when you have done it a few times, this will be blisteringly fast. So maybe for a much bigger precipitate than this, even with much harder problems to solve, might take something like 3 or 4 minutes. Yes, so that's kind of our result. And now, important as always, is to save, and this time I will save it as review. So before I go ahead and show you how to export the data, I just wanted to show you a little bit what you can do with these overlays. And let's say here that in this overlay tab there are some filters you can select or deselect. And typically for these images, we don't want to actually label the matrix because it's obvious. So I can turn off the matrix, and we see here that there are actually, these were the 2 columns we dabbled with in the beginning, which is now labeled as being part of the precipitate. And this can also be changed. There is if you go to selected column, there is one called precipitate column. If I uncheck that, it should go away once we reapply the filter. So this is how you can check that the precipitate is correctly labeled as precipitate. And this doesn't really matter for the visual overlay, but internally this can matter if you want to use that data for when you are exporting it for something. And you can take away this, and maybe you want to just look at the silicon network, you can do that. And I think one of the more fun ones is to look at only one plane. And yeah, it's all fun and games. And so now that and it's a bit buggy but never mind that. and so now perhaps you want to export this somehow. There are two kind of intended use cases for this. The first one is to actually generate these overlays, and you know, the graphics here are not very beautiful, they're kind of basic, and maybe you want to do like something better and more fancy for your kind of publications stuff. Then you can go ahead and go to the export button, and there are three options here, and one of them is scalable vector graphics, SVG. And when you export this as a SVG, you are able to make a SVG file where all this information is layered. So all the information is layered in silicon or aluminium, and you can kind of edit all of those graphical elements in kind of one go, in exactly the colors and you want or something. And there are some options when you are exporting. So remember from the species dictionary that there are two ways to categorize the species. The one is of course atomic species, which we all know, but the other is advanced species. Advanced species is just a term for, you know, the silicon 1, silicon 2 categories and stuff. For now I'll just take atomic species. Here you have a radius factor, so the radius of each graphical label will be scaled by the atomic radius with this factor. What we are looking at here is a factor of a half, which I think is probably the most reasonable. And you can actually include the graph, the atomic graph, if you like. This again will be layered, so you won't see the graph, but if you like turn off all the other things, then the graph will appear, and you can edit that as well. And you can include the image or not. Actually, this doesn't work to include the image, but with this box checked, it will draw a rectangle for where to place the image. And then you can also filter stuff. You can, you know, exclude columns in the matrix. You can, yeah, some things here. And here is our, you can recalculate the graph before. If you have done a lot of changes, then this might be a good idea, but if usually it can be left off. And you can just go ahead and click export and, you know, save it somewhere. And once you have done that, you can, you know, edit it in any SVG software. Like I usually use Inkscape, but there are many options. And the other intended use case is something I have not been working with, is that you can export the raw data with positions and you know, species. You can select many different attributes to append to that data, and it will make like a CSV file of it. And this is what you would do if you are using some other software which you want to analyze this structure with. Let's say you want to have the 3D structure of this in lamps or something. Then you would again, you will go to export, and you would select the comma separated values. And here you can actually choose to do only the current project, or you can actually enter like several. For now I'll just do this. If you want a 3D model in lamps, it only makes sense to have one project exported. But the reason for having a list is, let's say we want to do statistics for a bunch of images that you have collected from a area in a specific samples, and you want to do some statistics on that. Then you would like export the data from all the images you have. But for now I'm just gonna do this. And then here are some options, attributes which you can add to the data. Some of these are kind of a bit outdated and deprecated, but I think the most important one that people will most likely want is I, which is like a unique index. And then perhaps you would like let me see here if I can find it, the spatial coordinate, and spatial will give it in units of nanometers, and it will be, or picometers, I don't recall, but it's relative to some random point. And the other core x means image coordinate x, so that's the pixel coordinates, or relative to the pixel coordinates, but mostly you'll be using spatial coordinates. So, and you would add the y coordinate as well, and maybe even the z coordinate. If I could find it, that would be a really nice spatial set. And of course, probably you would like the atomic species, and maybe you would like some normalized average gamma. That's like a measure of the brightness of the columns. Maybe you would like that, and that is typically what you would get. So that is in fact like the output of the software is the actual three-dimensional coordinates and the atomic species. And there are some other things here that are related to the actual structure, but that's more, I'm not sure how you would use that necessarily. So then you would go next, and again you have some filters, and you have this option, and then you would just export it, and you have a CSV file which you can parse. You can like write scripts to parse it into whatever format or whatever needs you have. Yeah, so that was a not so quick start tutorial, but hopefully it should be enough to enable you to try it out and see if this is relevant for your work. So thank you.